Good evening, my dear students and friends. Myself, Dr. Ravikant Pathak, and I'm going to start your video lecture class. In the last video lecture, we have talked about the digestion of food, and in this video lecture, we will discuss about the bile juice as well as the digestion takes place in the small intestine. Okay, so here first of all, we are going to discuss about the bile, that means bile acid or bile juice. Okay, so if you read, so it is released into the duodenum. We know very well hepatopancreatic duct, because hepatic, so some hepatic ducts which carry this bile juice, okay, which bring this bile juice and open into the duodenum, and pancreas they also brings the bile juice and open into the u-shaped duodenum. Clear? So that if you read this statement, so this are given. Okay, so it is released into the duodenum, contains bile pigments. So these bile juice have the bile pigments. Okay, and these bile pigments, what they have, the bilirubin and biliverdin. Okay, and what is the bilirubin? Bilirubin, you know, RBC cells, that means they have the fixed lifespan. So 120 days considered as a RBC lifespan. And when RBC become degrade, because after when die, so there is the degradation takes place, means catabolic pathway. When catabolic pathway, that means degradation of RBC takes place, that time this bilirubin forms. Okay, this bilirubin forms, and this bilirubin is considered as a, this bilirubin further degraded into the biliverdin. And bilirubin is the yellow color pigment. If proper not degradation of takes place bilirubin, then here the deposition inside our bodies, this yellow color pigment deposition takes place, and it will cause a disease which is known as the jaundice. Okay, so this is the essential process, because for the formation of heme, so bilirubin converted into the biliverdin, and biliverdin forms the heme. Okay, so when bilirubin will not convert, and you know bilirubin is the yellow pigments, when their concentration increases inside the body, it cause a disease, that means you can say jaundice. Okay, and they have given the bile salts. Okay, so if they have given the bile salts, and you know bile is the derivative of the steroid hormone, and steroid hormones also derived from the cholesterol. Clear? Because every steroid hormone they have derived from the cholesterol, and bile term also derived from the cholesterol. Okay, so then they have the given cholesterol as phospholipid, but no enzymes in the bile juice. They have no enzymes, you should to know. In case of the pancreatic juice, they have they have most of the enzyme, but in case of the bile juice, no enzymes found. Okay, and how many types of the bile acid found? So four types of the bile acids found: cholic acid, deoxycholic acid, chenodeoxycholic acid, and lithocolic acid. Okay, so these four different types of the found, in which two are predominant. Okay, in which two are most abundant: cholic and chenodeoxycholic acid. So these two are considered as a most abundant bile acid. Okay, most abundant bile acid. And here one statement is also very important. One statement is the very most important. So cholesterol excretion generally performed by the formation of bile salts. Okay, bile salts are bile juice, because cholesterol is considered as a carrier of the fat molecule in blood. Okay, cholesterol considered as a carrier of the fat molecule in blood. Okay, next one, bile helps in the emulsification of fats, that is breaking down of fats into very small micelles. Micelles, you know what is the micelles? Micelle is a special type of structure. You already studied in the chemistry. So that is the washing of clothes. And you know during the washing, what we will generally use? We have generally used the detergent, and this detergent have the one enzyme which is known as the lipase. So similarly, lipase, you know, they have the lipase, and this lipase involves in the washing phenomenon. So when you will wash the clothes, so that time they forms, because why dust came, or why your every clothes become dirty due to the deposition of fat molecules. Okay, and when you will use this detergent, so this detergent with the fat molecules bind and remove, and your clothes become clean. Okay, your clothes become clean. That means during the washing mechanism, what that means, washing of the clothes, so you can see there is the formation of micelles takes place. What is the micelles? You know, lipid molecules. Lipid molecules have the two region. One is called the head and another is the tail. So head is always hydrophilic and tail is the hydrophobic. Okay. So here it forms a ring-like structure where outside they have the head molecules, that is the polar molecules. Okay. Outside they have the head molecules or the polar molecules, and inner side they have the non-polar, that is the tail region. Okay, and these inner sides they have the form like this structure, and this special structure is known as the micelle. Okay, micelles are micelles, and these inside they have ability to bind with the fat molecules because hydrophobic, and you know very well fat also is the hydrophobic because lipid means oil is the immiscible in water. Okay, so this is the micelle structure. Similar micelle structure, they also form one special structure which is known as the liposome. Liposome is the same thing, but here liposome, this is the infolding of the plasma membrane, because you already seen plasma membrane. Their outer side means extracellular and intracellular. They have the hydrophilic, means that you can say head region. You already seen. So suppose that this is the lipid molecule. I am drawing here, so you can see this is the lipid molecules, and these lipid molecule here they have the head region. So sorry, this is the tail region. Okay, this is the tail region, and here also tail region. Okay, so similarly, suppose that this is the ECF, extracellular fluid, and now same thing also here you can see in this figure. So then here this is the head molecule. So it means intracellular fluid as well as intracellular as well as extracellular. They are provided with the hydrophilic tail. Okay, hydrophilic region, hydrophilic head, and tail region. So they have the hydrophobic tail. Clear? So they have the hydrophobic tail, and if this moves, if it moves like this, then it will forms. Okay, it will forms a special structure which is known as the liposome, and this liposome we will also discuss in the biotechnology. This is used for the drug delivery on gel delivery method. Clear? So this liposome, so this is like similar to micelles, but they have the two layer. If you see outer hydrophobic and outer hydrophilic and inner hydrophilic, but in middle they have the hydrophobic region. Okay, so this liposome is used for the drug delivery as well as gene delivery purposes. Clear? So here bile helps in the emulsification of fats. What is the emulsification of fat? Because fat is the very big and large molecules, and one also important property of the fat, so fat never will forms the biopolymer, because you already seen. So protein also forms. Okay, protein also forms, carbohydrate also forms, and nucleic acid also forms, but biomolecules only the fat never form the biopolymer. Clear? Next one, bile also activates the lipase, because I had already told in the detergent they have also enzyme lipase, and you know very well lipase involves in the degradation of lipid molecules, that is the lipolysis. Okay, the secretion of the brush border cells of the mucosa along with the secretion of the goblet cells. Goblet cells means mucous neck cells. So goblet cells as well as brush border of the mucosa, they secretes the one juice substances which is known as the Succus entericus. What you will say? This is the Succus entericus. Clear? So this generally secreted into the duodenum, and this secreted by the brush border mucosa as well as goblet cells. So brush border mucosa as well as goblet cells both secretes one substance which is known as the Succus entericus, or simply you can say intestinal juice. Okay, if you will see next, the mucus along with the bicarbonate, we have already known what is the function of that means bile salts. They also increases the pH. They also increases the pH value. Clear? Because we have seen in case of the stomach there is the pH very low, but when it will pass inside the duodenum, so their pH increases. How it will increase? So here you can read with these statements. Okay, the mucus along with the bicarbonate from the pancreas protects the intestinal mucosa. They also protects, this mucus protects from the intestinal mucosa from acid as well as provide an alkaline medium, because this mucus secretes and this mucus generally provide the alkaline medium. Okay, and for enzymatic activity, so alkaline medium pH 7.8, and also for enzymatic activity, because enzymatic activity here favors alkaline pH, but in case of the stomach they favors the low pH. Okay, and submucosal glands, that is the Brunner glands. Brunner glands I had already told. These Brunner glands, they have also a special types of the cell which is known as the Paneth cell, which is known as the Paneth cell, and these Paneth cell involved in the secretion of the antibacterial enzyme like lysozyme, and it also help in this phenomenon. Clear? So that means you can say Brunner glands, that is some mucosal glands, and one thing also intestinal mucosa, they secretes the mucus, and this mucus provide the alkaline medium, and for the enzymatic activity it is also essential. Okay, next one, complete digestion of food takes place in the duodenum. This is the very important sentence. They already asked one time NEET examination. So where the complete digestion takes place? So complete digestion takes place in the duodenum. I had already told, in mouth only 30% carbohydrate digest. In case of the stomach, only protein digest, and here in case of the duodenum, everything, protein, vitamin, sorry, protein, nucleic acid, and carbohydrate, and carbohydrate, fat, everything digested here. One thing, one term is very important, the fully digested food in semi-fluid nature. So the fully digested fluid is semi-fluid in nature, and this structure is known as the chyle. We already studied in case of the stomach there is the chyme, but here it is called the chyle. And what is the chyle? When the fully digested means complete digestion process, and this after the fully digestion it forms the semi-fluid in nature, semi-fluid like structure, and this structure is known as the chyle. Okay, and these final steps in the digestion occur very close to the mucosal epithelium cells of the intestine. So we know very well, so they have the mucosal epithelial cells, and here that means final steps of the digestion occur. Okay, final steps of the digestion occur near to the, that means very close or near to the mucosal epithelial cells of the intestine. And in case of the large intestine, no digestive activity, only absorption takes place. And what substance absorbs? So here you can see there is the absorption of some water, minerals, and certain drugs. In case of the large intestine, absorption of some water, minerals, and certain drugs takes place. Okay. Next one, the undigested and unabsorbed substances called feces. Okay, and these feces enters into the cecum, and after cecum, that is the large intestine part, and cecum of the large intestine through ileocecal wall. They have the ileocecal wall, and these walls prevents the, what is the function of wall? This wall prevents the backflow of the fecal matter. So it is the only one direction pass. It will not reverse back. Okay, so this function performed by the ileocecal wall. Okay, next one, the muscular activities of different parts of the alimentary canal, so can also be moderated by means, that is a regulation mechanism. So this regulation mechanism of this alimentary canal, it controlled by the both local as well as central nervous system. Means, so local, that means you can say peripheral and other things, as well as central nervous system, they involves in the controls muscular activity of the intestine or alimentary canal. Hormonal control, we know very well, so they have also secreted GIT, gastrointestinal tract hormone. Okay, so here hormonal control of secretion of digestive juice is carried out by the local hormones produced by the gastric glands, G cells. I already told, gastric gland G cells, they also secrete hormone, and intestinal mucosa, that means intestine also secretes the hormone. Okay, and now we are going to finish your class, and in the next video lecture we will talk about the absorption of the digested products. Okay, and thank you for your patience and cooperation, and I'm expecting with you, so everyone kindly subscribe my channel and hit the bell icon for regular updates. So thank you.